Good morning, everyone. It's our pleasure to have you here today for our roundtable discussion on the amphibian crisis. I wanted to start off by welcoming one of our honored guests, Mr. Abi Said from the Panamanian Embassy, sitting here in the front row. And it's also my pleasure to introduce our good friends and partners on this initiative, which we're calling the Panama Amphibian Rescue and Conservation Project. The first person I'd like to introduce was very important in having helped us get this program started. John Barry, about a year and a half ago, went to the AZA conference and gave quite a speech in front of the assembly there, and he basically called to order all of the zoos to work together to help rescue amphibians. And so this was really a call to action, and he's a very passionate speaker, and it was very effective. But then he had to find some willing partners to stand with him to actually launch a program where zoos could show leadership in this area. And he reached out to Bob Chastain, who is the president and CEO of the Cheyenne Mountain Zoo. Bob, would you please stand and be recognized? Thank you. And Bob made a very solid commitment to us for 3 years and was really the first one to come on board. And not only that, he helped us to recruit additional directors to stand with us. And so we're very grateful to Bob for his leadership in that way. I'd also like to represent or recognize the Defenders of Wildlife, which is one of our partners. And Cindy Hoffman is here representing Defenders. Is Cindy here? Thank you. Next, I'd like to recognize John Lenihan. He's the president and CEO of Zoo New England. Thank you, John. Also, the general director of Africam Safari in Puebla, Mexico, Amy Camacho. And a good friend and colleague, Dr. Biff Birmingham, who's the director of STRI, the Tropical Research Institute in Panama. And also Adrian Benedetti, who's the director of the Summit Park in Panama. And finally, one person who's not here today to join us, but who's a really important partner, is Rick Barangi, who's the director of the Houston Zoo. And Rick has been a leader in getting zoos involved in amphibian conservation now for several years, and so thank you to Rick. Amphibians are an amazingly beautiful and diverse group of organisms. There are supposedly somewhere around 6,000 species now that have been identified, but we're continuing to discover new species all the time. There was an article out last week where scientists using DNA barcoding identified another 200 species of frogs in Madagascar, and that doubled the number of known species alone just in that one country. The scientific data that we use to look at threats to different species is pretty complete in about 2/3 of the cases of the 6,000 species of amphibians. Unfortunately, that means the threat levels show us that about 42% of all of the species of amphibians are threatened with extinction, according to the IUCN. And this is a pretty astounding rate. It's about 4 times the number of species of birds that are threatened, and more than almost twice as many as the number of mammals. Since 1980, more than 122 species of amphibians have gone extinct. And this compares to just 5 species of birds and no mammals that we're aware of. So this is a very alarming trend, and this is where the term amphibian crisis comes from. In fact, we may be witnessing one of the greatest extinction crises of vertebrate organisms that the Earth has ever seen. A deadly fungus, the chytrid fungus, is a disease that was first discovered and studied here at the National Zoo. We now know, and it's widely recognized, that this fungus is responsible for this enigmatic amphibian decline that's been witnessed around the world and has led to the extinction of species like the gastric brooding frogs of Australia, the Monteverde golden frogs of Costa Rica, the golden Coqui frogs from Puerto Rico, the Panamanian golden frog, and even the Wyoming toad here in our own country. So today, what we've done is we've assembled a truly distinguished group of experts who are going to tell you something about the current plight of amphibians, and also about the direct cultural and ecological and biomedical value that amphibians bring to humankind. And to me, most importantly, we're going to be telling you about what kinds of action steps that we are going to be taking to help solve this crisis for a number of critically endangered species in Panama. So our first speaker today is Dr. Karen Lips. She's the incoming director of the graduate program in sustainable development and conservation biology at the University of Maryland. And Dr. Lips is one of the world's foremost experts in amphibian conservation and ecology. She's worked for many years throughout Latin America and has personally witnessed the devastating impact of amphibian chytrid disease as it swept through Costa Rica and western Panama. By analyzing the patterns of decline among species and across many sites, Karen is helping us to predict which species are likely to go extinct in the future and to help us develop data for prioritizing the conservation and research activities that will help us to alleviate the population decline. So please welcome Dr. Lips.
Well, thank you all very much for the invitation to come and talk about some of our work, and in the hopes that the zoo and the Smithsonian and all the different partner groups can hopefully do something to save amphibians. My story, I'm sort of one of the observers. I'm the one in the field watching these frogs die in front of my face, and initially I was working at such remote sites that most of the species I found were new and undescribed. And I was thinking about this the other day, and I realized that at this point, I may have seen more species go extinct than describing new species. And that's something I think pretty unusual in the world, to actually be able to say that you've seen many species go extinct in front of your face. And part of that is because of the places that I work are quite remote and have many endemic species. So as we just heard, the amphibian story is really different. It's different in terms of the scope and the scale of extinction, much different than sort of our typical endangered species, where in general it's one species that's either over-harvested or affected by habitat loss, or maybe a region that is being affected by habitat loss or some sort of extraction. But the amphibian story is very different. What we're talking about now is a shift in paradigm to something that is more regional, global, where the problems and the threats, like this fungus or climate change, cross geopolitical boundaries and make it much more difficult to come up with some sort of plan to save the affected animals. So what we're seeing now with the amphibians is the loss of an entire class of organisms. And with the loss of coral reefs, also due to things like disease and climate change, we're seeing the loss of entire ecosystems. And unfortunately, we're hoping this is not the beginning of more similar stories to come for other groups or areas. But this little schematic sort of gives us the idea of what's going on. We're seeing now the loss of all sorts of unique species. And what we're left with are fewer species, but also fewer individuals of those surviving organisms. We just heard a short synopsis of the problem. Again, we're talking thousands of species of amphibians. Most of those species occur in the neotropics, from the border of Mexico down to the tip of South America. And this is where we're seeing some of the greatest losses. And so we really are seeing the loss of an incredible diversity of this entire class. And again, what I'm going to be talking about, and all the talks today, are really focusing on this disease and its problems. This is from a great website by Dee Olson, who's gathering data through the park system. And each dot represents a place where BD, this fungus, has been recorded. And you can see that it's covering much of the map. We can see North America and Central and South America in particular have quite a number of sites. There seem to be some areas where it's either not yet been described or might not have gotten in. The good news is places like Madagascar have been searched and are so far clean. So something like our meeting today is a really good effort to try and understand what we can do to prevent the spread of this fungus to places where it is not yet present. So just to summarize that map, BD is almost everywhere at this point around the globe. It's been reported, again, from that same database, from many, many different countries. Where we've tested it in many animals, most species can be infected by this fungus. And at this point, I think we're up to almost 200 different species have tested positive. So this is a major problem. The key points about this are that it's global. It's very recent. This is a recent problem, only happening in probably the past perhaps 20 to 50 years. And we keep sort of moving that timeline back as we do additional work out of museum specimens. And again, it's quite widespread. Where the disease is present, we do see the extinction of species, and it tends to be in quite remote areas with many unique endemic species. And where BD has been a major problem, there's very little good news. There's almost no good stories of recovery of populations or return to normal or typical levels. In some cases, populations persist, but they don't tend to rebound to high levels, at least in the neotropics where I've been working. This fungus has been described as an invasive species. It now has an official status, which helps us now legally try and mitigate its impacts through the policy. And it has been introduced globally to many different places of the world, and I'll talk about some of the introductions that we have data for from Central and South America. And what's really important is that temperature and moisture are really the key things that determine how fast this fungus grows and how well it grows. And that really will sort of be a major theme, I think, today. And unfortunately, it persists in the environment, and that's where the zoos really become so critical, because once it arrives to a site, it kills off many of the frogs, it still persists in those few individuals that do remain. And so until we can come up with a way to get it out of the environment, any sort of reintroduction plan is going to be a difficult story. And again, it infects many different species. So I'm going to give a quick description of sort of the ecology of BD. Reid's going to talk a little bit more about what it does to frogs. Here we have a great amphibian habitat with a tadpole. Here we have the zoospores. These are little zoospores that are part of the life cycle of the fungus, and they like to live in cool, moist, aquatic habitats. And as far as we know, it's amphibian specific. It only infects amphibians, but it infects many different species of amphibians. And so we have it infecting these two different species here, and in the upper case now, once the zoospore infects the frog, it creates a zoosporangium where it then produces additional zoospores that either reinfect the environment or reinfect the frog itself. And so if a frog gets enough reinfection, it will eventually die when it reaches a critical limit of too many zoospores. And frogs can pick it up then out of the environment, or when they go to the habitat to breed, they can pick it up from other individuals as well. And so again, temperature is really critical, and in the end, what we tend to see at the sites that I work at are sort of two general responses. That is, some species go extinct. They're either rare to begin with, few in numbers, or occupy habitats that really allow that fungus to persist and sort of grow so fast that eventually most of the individuals get infected and die. But other species are able to persist, and it's these where we're really looking for clues as to how it is that some can persist. And there's good evidence that there are a variety of defenses that the amphibians have. There are certain ecologies or microhabitats that are not real good for the fungus, and it's probably a combination of these things that allow some species to persist while others do not. And so this is sort of our group, working with Louise and Reid and many other people, of the different types of research that we're doing, looking at the amphibians, looking at the fungus, and looking at the environment that they share to understand under what conditions can a species persist and where will they disappear. So I'm going to real quickly talk a little bit about some recent work that we're doing. I've spent all my research career in Central America. I started out in Costa Rica doing my PhD. And eventually moved to Panama because I ran out of frogs. There were widespread mortalities in Costa Rica, and unfortunately, Panama is on its way to losing most of its upland frogs as well. This is a map that shows the incredible amount of mountainous areas of Costa Rica, and also, to a lesser extent, here in Panama. And these then are reported areas of massive die-offs or declines of amphibians associated with the chytrid fungus. And what you can see is they make a nice or a sad wave of epidemic spread. And this has been held up pretty well under our predictions. It gives us the ability to predict where it's going to go next. BD has now crossed the canal and is moving into eastern Panama. And this is now where I work. I work in these little upland areas in eastern Panama, and we hope that it will somehow stop or not move through those areas. And again, it's these cool upland areas that really seem to get hit hardest. It is present in the lowlands, the fungus. It doesn't seem to have quite the significant impact on the amphibians as it does up in the mountains, where it's sort of the perfect incubator for BD. And so most of the stories I'm going to talk about today are from these upland sites where the impact is greatest. So I'm going to talk first about one case study, a site that we worked at for 8 years. Every year we'd go down and spend several months at El Coqui in central Panama. And at this site there were 65 species of amphibians that we knew about. And the pictures here are a great example of the incredible diversity and just spectacular appearance of these animals. BD, we were sampling for many years, and we could say conclusively that it was not there. And then one day, we got our first positive hit. The next week, we had the first dead animal, and for the next 4 months, there was a huge, massive die-off. Within 4 months, we lost over about 60 to 70% of all the amphibians at this site. In terms of species, we were missing species within 2 or 3 months of that initial infection, and the number of species continues to go down over time. Most of the species at this site were found either dead or infected with the fungus. Those that were not were sort of so rare that we didn't ever really find them at all. And it infected all different habitats. We had species that we didn't know existed at this site because they lived up in the canopy or they lived underground. And in some cases, these animals would fall out of the canopy because they were sick and dying, or come bubbling up out of their burrows underground because they were sick and dying. And again, I can't impress the speed at which this happened. I mean, it was just absolutely incredible. And we only found 500 some dead frogs, but you can imagine this is a very small fraction of this very small valley in one little site in Panama. So the actual number was obviously much, much, much greater. This is sort of the data. These are our surveys from when we first, moved there. Uh-oh, no, I'm stuck. In 1998 through our last survey there in 2005, and this is just an average estimate of the number of frogs that we would find along our stream transects. I can't go backward. Anyway, both of these slides show both the number of species and also the number of individuals that disappeared backwards. I know, I had the same problem. There we go. So basically you can see as soon as we find BD at this site, there's an instant decline in not only the number of animals that we find on a transect, but also the number of species. And again, this is just the average number, but there have now been the loss of many species at this site. Over 40% of the species are now missing, and those that were only found at this site, we presume, have now gone extinct. This shows you then those species and individual species decline. So each line is one particular species at this site, and this is sort of their estimate of how abundant they were before the BD hit, and then what they're like today after the decline. We can see that all species go down. Some go down faster and more than others. This is the Panama golden frog, and that is this dot here. It was one of the most abundant species, and it is now gone from this particular site. So it really is rapid, and it affects all the species there. This is a representation then of all the diversity of amphibians at this site. This is a paper I'm working on with Biff Birmingham and Andrew Crawford actually, where Andrew went through and barcoded all the different species at this site to really capture the true diversity. And it turned out I missed 10 different species that were not known because they were indistinguishable from the other species. So here we have that tree of diversity of all the species, including those that were not known, listed in purple, and in red are those that now are missing. So we can see here that this tree of diversity is much smaller than it used to be. That is, it's occurring all along the tree. We really are losing a wide range of different ecologies and species and diversity of our amphibians. This adds to that loss, not just those that have completely disappeared, but also those that have declined by more than 60%. And again, if we now just look at the black lines are those that we think are okay, stable, have not gone down. And you can see the number of black lines are very, very few. Almost all species have declined to some degree. Some are completely gone, and very few actually persist at typical levels. So again, here we are back at this map to sort of remind us where we are. I want to now sort of briefly sort of take a step back and look at what's going on for all these sites. Here is El Coqui here, 2004, that I just told you about. Well, what happened to all the amphibian faunas of these other sites? Well, this is a similar figure. This is not an individual species, but in this case, these are the entire amphibian fauna for all these different sites, starting here in Monteverde in Costa Rica, La Selva, southern Costa Rica, and into then central and western Panama. And again, you see the same pattern. In terms of the number of species, it was quite high before BD arrived at each of these sites, and has gone down rapidly and pretty significantly following the introduction of BD. There's a lot of text. All this does is sort of summarize for the big picture for all these sites. If we sort of look at them as a group, what we see is we've lost about 50% of the species across that region of Costa Rica and Panama. And what's most disturbing is that most of the species that have disappeared throughout this region are the endemic species. There were 61 endemic species. 42% of all those different species were only found at one site. And it's those species that have disappeared from across this region. So what this means then is not only are we losing species, but we're losing the really cool, unique aspects of the diversity at these different sites. And so you look at this region today, and it's homogenized. The amphibian fauna across this region is more similar today than it was in the past, where each site had its own unique elements. Those unique elements are what are disappearing from those sites. So these are our non-random extinctions. We're really losing those things that are found nowhere else in the world. And BD, this exotic introduced species, is now driving the pattern of diversity throughout this entire region. And given the patterns that we've seen elsewhere in the world, it's probably happening in other parts of the world as well. These are just a couple of the frogs that I watched go extinct from some of my sites. You can see they're incredibly spectacular and beautiful, and we're pretty sure that these are gone for good. Just a handful of the many. And unfortunately, this pattern has held up elsewhere. This is some of our work from South America. Here's our map from Costa Rica and Panama. Here are similar waves of movement of BD that we've predicted throughout the Andes of South America that happened many years ago. So most of South America has already been affected, and it's too late to save many of the upland species at this site. But what that leaves us then is this little bit of Panama. This is the last remnant healthy amphibian populations in all of Central and South America in terms of these upland sites. Okay? It's not just Costa Rica and Panama. This has already happened through Mexico, Honduras, Guatemala. It's already happened through South America. These are 5 sites that we're working at. We've tried to get in there and do some expedition work, again with Andrew Crawford and Roberto Ibanez. We now know that this one is positive. And we can see that here is BD in South America. Here it is here near the canal. And we're down to these few upland areas where we believe these sites to be healthy and uninfected. Of course, we predict that it's only a matter of time, perhaps 5 years. If it continues at its typical rate of movement of about 22 kilometers a year. So unfortunately, the status of amphibians is really bad. It's probably much worse than we even appreciate, given all those cryptic species, given the loss of regional diversity, and given the true diversity that's out there. And it's going to get worse as it continues to spread. When it hits Madagascar, when it hits Asia, these other areas will be affected as well. So unfortunately, the news is not great from the field, but I'd like to thank you all again for the invitation to come and answer any questions. Thank you.
So we'll be doing our questions and interviews at the end of all the presentations. Thank you very much, Karen. Our next speaker is Dr. Louise Rollins-Smith. She's an associate professor of microbiology and immunology at Vanderbilt University. Dr. Rollins Smith is one of the leading researchers in amphibian developmental and comparative immunology. Currently, her research focuses on the development of the immune system and the nature of innate immune defenses in frog skin. Understanding the immune defense mechanisms of amphibians has taken on increased importance in recent years because of the urgent problem of the global amphibian declines, but it also highlights the importance of amphibians to humans, demonstrating that amphibian skin secretions can potentially inhibit HIV transmission to T cells. Please welcome Dr. Louise Rollins Smith.
Thank you. Let's see if we can pull up my PowerPoint real quickly. So Karen's done a great job of sort of introducing you to the amphibian crisis in Central and South America. It's also true, it's happening all over the world, including Australia, and I'll show you some images of Australian frogs that are affected as well. So my role today here is really to sort of address a question that comes up often when we talk to reporters or when we talk to family and friends about the amphibian decline problem. And that is, why should we care? Why do we care about amphibians? What's their importance to human beings? Why do we really want to know, why do amphibians matter? And I think for a biologist, it's a really simple question. Maybe more complex for other people, but it's really simple to me. When you look at an image of this Fleischmann's glass frog from Panama, this is a highly evolved, complex creature worthy of our interest, worthy of the interest of biologists. And in my view, the world would be greatly diminished if we lost all of these beautiful creatures. And that's sort of illustrated by another quote from the naturalist and biologist Archie Carr. He said that frogs do for the night what birds do for the day. They give it a voice, and that voice is varied and stirring thing that ought to be better known. These are just a couple of images of another amphibian from Panama, the lemur leaf frog, some of which are just here in the collection. Hylomantis lemur, and an image from the orange-thighed frog from the rainforest of Australia, northern Australia. Again, beautiful images of amphibians. But there are other points that I'd like to make about the importance of amphibians to humans. There is their role in the ecosystem. This is an image from Panama of one of the locations where Karen's done some collecting and someone from my lab has done some collecting. And it's important to remember that the adult frogs are primary consumers of all kinds of invertebrates, especially insects. They're great little bug eaters. Tadpoles are consumers of algae and other plant matter, so they sort of control the growth of some plants and algae within the streams. And both the tadpoles and the adults become food for other groups, including fish, amphibians, snakes, and birds. So they're an integral part of many ecosystems, and we really don't have a good, clear understanding of what will happen if they disappear entirely from these ecosystems. Another important role for amphibians as indicators of changes in the environment. There's evidence that when there's too much nitrogen and phosphorus in an aquatic system, it provides a rich environment for growth of algae and plant material that results in overgrowth of snails. The snails are intermediate hosts for a trematode parasite that results in limb deformities in frogs. So increased limb deformities in amphibians is probably an indicator that there's a change in the environment. There's some evidence that the agrochemical atrazine may alter the sex development in amphibians, and from our own work, there's some evidence that the pesticide carbaryl may inhibit the release of defensive antimicrobial peptides in amphibian skin. Amphibians historically and currently play an important role in biological research. Their biology is very similar in many respects to the biology of all other vertebrate groups, including our own. The amphibian eggs and embryos are large and easily manipulated. These are some images from some experiments that I did some time ago, transplanting a region of the embryo that forms the blood in an amphibian. Here's a little transplanted embryo with a blood-forming region transplanted, and a few days later, the embryo entirely healed. So currently, developmental biology studies rely heavily on amphibian model systems. First studies of cloning were done with amphibians, and there have been at least 7 Nobel Prizes based on research done with amphibians. So the last point I want to make is the possible role that amphibians, the products of amphibian skin may be useful in development of certain kinds of drugs or other therapeutic agents for treatment of human diseases. Many amphibians, many frogs and toads, have specialized glands in the skin called granular glands. So this is a granular gland. It's packed full of little vacuoles that are full of defensive peptides that are defensive against predators or defensive against microorganisms that may injure the skin. You can sort of think of those skin glands as containing little packets of poisons or as a sort of a first aid kit. So if the frog skin is injured, it has a painkiller as well as antimicrobial peptides to sort of sterilize the wound. Those antimicrobial peptides, this is my cartoon of an antimicrobial peptide ready to hammer a pathogen that comes along. They're capable of killing bacteria, both gram-negative and gram-positive, including those that are called methicillin-resistant Staphylococcus aureus. This is a bacteria that lives on the skin and in the nose of many people and is very worrisome because it causes sepsis and very serious illness, and it is currently becoming more and more resistant to all the common antibiotics. So amphibian skin peptides, some of them are very competent in destroying methicillin-resistant Staph aureus. In addition, many of these peptides are capable of inhibiting viruses, including HIV, and I'll say a little more about that. They inhibit fungi, including the chytrid fungi, and we believe that the repertoire of antimicrobial peptides in the skin of amphibians, many species, is part of what enables some of them to persist with the chytrid and to kill protozoan parasites. This is an image of bacteria that have been treated with antimicrobial peptides. You can see that the contents of the bacterial cells are coming out of the bacterial cell, indicating that it will die as a result of the antimicrobial peptides. Because we knew about the properties of antimicrobial peptides, we thought they might be effective in inhibiting a virus like HIV, which has an envelope, which is very much like a cell membrane. So in collaboration with HIV virologists at Vanderbilt, we decided to test some of the peptides that we had been isolating from amphibian skin. And it turns out that we tested only about 13 or 14 to begin with, and we found at least 3 that were very effective in inhibiting the virus. This is low concentrations, and as you increase the concentration up to what is a fairly small concentration, 10 micromolar, all the viruses are inhibited. So this was the best peptide, and these 2 were also quite effective. Serin 1.9, Serin 1.1, and Serin and maculatin 1.1. These effective peptides all came from Australian frogs, the red-eyed tree frog, the green tree frog, Latoria caerulea, also called White's tree frog, and the green-eyed tree frog, Latoria genimaculata. These are all short peptides. They're easily synthesized once the sequences are known, so you don't have to go to the frog to get the peptide. You can actually have a chemist synthesize it, and the chemically synthesized ones are equally effective as the natural peptides. This is another way to visualize that. In this experiment, the viruses were modified so that they expressed a green fluorescent protein, and in this case they're being taken up by an intermediate cell called a dendritic cell that passes it along to the T lymphocyte. So in the bottom panel, this is a dendritic cell that has picked up viruses ready to transfer them to T lymphocytes. It's been treated with the ineffective control peptide, whereas in the upper panel, the viruses were pre-treated with the effective antimicrobial peptide, serin 1.9. All those viruses were destroyed. There are no green viruses being picked up by this dendritic cell. So finally, I'll just leave you with, just remind you of just a few of the potential molecules that have been isolated from amphibian skin that could be beneficial in human health. Meganin has been isolated from the South African clawed frog, Xenopus laevis, and it is an antimicrobial peptide that has been proposed as a cure for diabetic foot ulcer. Epibatidine is an alkaloid isolated from the skin of an Ecuadorian frog that is a very powerful painkiller and is non-addictive because it binds to it uses a different biological pathway than the morphine addictive painkillers. Another antimicrobial peptide from the waxy monkey frog phylum Medusa sauvagei, dermaceptin, is one of those that is capable of killing antibiotic-resistant Staph aureus. We've already mentioned serin 1.1, which is one of those that inhibits HIV, and the fire-bellied toads, Bombina, as well as a number of other species, produce a small peptide, a 9-amino acid peptide called bradykinin that is one of the agents that can reduce high blood pressure. So in summary, frogs are beautiful, exquisite, complex creatures. They have an important role in ecosystems. They're good bioindicators. They have been important to biological research historically and presently, and they may hold secrets that will reveal compounds in their skin which are beneficial for human health. And with that, I'll turn it over to Reid.
Thank you, Louise. Our next speaker is Dr. Reid Harris. He's a professor of biology at James Madison University. Dr. Harris is a professor of biology at James Madison in Harrisonburg, where he teaches classes in ecology and evolution, and he conducts research on amphibian life history evolution, behavioral ecology, and most recently on disease ecology. His current research focuses on using amphibians' naturally occurring protective skin bacteria to control or prevent a lethal skin fungal disease. So please welcome Reid.
Well, thanks for the opportunity to talk to you today about can we use another one of amphibians' innate defenses in their own conservation. All right, as Louise just mentioned, we should care about the loss of amphibians for many reasons. They can constitute a really high biomass in certain areas. They're indicator species, and of course they can contain important pharmaceuticals that we can use as humans, in addition to being really evolutionary marvels. We should probably not forget that there are other threats to amphibians other than chytrid, such as habitat loss and pesticides. But of course we're here to talk today about chytridiomycosis, which is caused by BD. And it's been explanatory in explaining these enigmatic declines of amphibians in otherwise relatively pristine habitats, as Karen talked about. And we should also remember that chytridiomycosis is an emerging infectious disease, and we need to place it in context with this unfortunate list of other and many other emerging infectious diseases that we're seeing today at increasing rates. So what to do about the BD, getting back to BD. A vaccine is being worked on by members of Louise's lab. That's probably a few years down the road. We can treat individual frogs with antifungal drugs. That can be helpful in laboratory situations. There are protocols for humans for us to limit transmission or moving BD around the planet. That's very important. And of course, survival assurance colonies and reintroductions is going to be very important as well. However, there's been relatively little research on how to manage this chytrid fungus in nature. So we're looking at this question, do amphibians themselves hold the key to their recovery? So I was actually, a little historical note, working on this species, the four-toed salamander, the females of which brood and protect their eggs. Now, there are other types of fungi other than the chytrids that if this female were to desert her nest, all the eggs would die. So we have fungus-free eggs and then eggs killed by fungus. This is other types of fungi. And we are working on, are the symbiotic skin bacteria, are they actually killing off the fungus and keeping the eggs relatively fungus-free? So then it occurred to us some time into this project that the chytrid is a fungus, and maybe the skin bacteria that we were working on as protective of the eggs might be protective of the adults against the chytrid fungus. All right, so our question was then, does the skin microbiota, the bacteria in effect, of the host, being the amphibians, inhibit the chytrid fungi? So here's a scanning electron micrograph of salamander skin, and you can see that there are various bacterial cells on the skin. So what occurred to us then, when the BD zoospore lands on frog skin, it doesn't really find a blank slate. It'll find the antimicrobial peptides that Louise talked about produced by the frog itself, but the zoospore is also going to find bacteria, and we know bacteria produce all sorts of antibiotics that might be anti-chytrid as well. So we were able to isolate a number of skin bacteria from amphibians. Here's a lawn of the BD pathogen on this Petri plate. Here's a streak of bacteria, which ended up being Lysobacter, that you can see is clearing out a big zone of the chytrid fungus. So it's killing off the chytrid. This species of bacteria was not able to do that. It leads to a lot of various lab work, although the student was actually very happy to be doing all this lab work. Yeah. So we found a number of different skin bacteria that are very effective, at least in these laboratory assays, of killing off the chytrid fungus. All right, so I want to talk briefly about an experiment that we just finished and just got published on working on the mountain yellow-legged frog out in the Sierra Nevada mountains, which is highly affected by BD. Can we add some of this anti-BD skin bacteria, and will that prevent or reduce morbidity and mortality caused by the pathogen? This is important because, much like in the Central American situation, this is in the high Sierra Nevada mountains of California. When the pathogen arrives in a population, you can get this almost entire mortality. Okay, so it was a relatively simple experiment. We had some frogs, this was in the laboratory, where we just added bacteria to their skin as a control, gave them a little bacterial bath, a little probiotic bath of Genthinobacterium lividum. Another group of frogs just got the pathogen alone. And then, of course, the treatment of most interest was we gave them the anti-BD bacteria first, then followed by the chytrid fungus. There's getting their little bacterial baths. Okay, so these were the survival results. Here's week of the experiment on the x-axis and percent survival on the y-axis. So, as you might expect with this species, when they were only just infected with a chytrid fungus, we got pretty much complete mortality. They were all dead, except for a few other ones that looked like they were on the way at 20 weeks. If we just add the bacteria control, we got perfect survival. And what we were most excited about was when we added the bacteria first, then infected them with BD, we also got perfect survival. And they also got similar effects on growth rate. We've been able to determine that this bacterium that we added produces this metabolite violicine, which we've shown in laboratory studies is highly effective at killing the chytrid fungus. So the overall conclusion then was skin bacteria produce these anti-BD metabolites which can kill BD and protect the frogs, and that we were able to bio-augment or add skin bacteria to the frogs, and that prevented them from having the mortality and morbidity that we usually associate with BD. And this paper just came out in the ISME journal, the International Society for Microbial Ecology. Okay, so this bioaugmentation in a lab study seemed to be successful with a frog species in North America, the high Sierra Nevada mountains of California. Can we extend these results to a neotropical species, in particular the Panamanian golden frog shown here? So these Panamanian golden frogs may be extinct in the wild. However, they're very healthy populations in a number of different places, including here in survival assurance programs. And they seem to be very susceptible to BD, which, as Karen mentioned, is endemic in the habitat. So that creates a problem. How can we put these? We've got this kind of paradox. We've got really successful survival assurance programs breeding up these frogs, but we can't just put them back into the habitat because they're susceptible to the pathogen, which is still there. So what we plan to do this summer at the Conservation and Research Center of the Smithsonian in Front Royal, Virginia, is repeat the experiment that I just went over with the mountain yellow-legged frog with the golden frog. Will they be protected from BD? How long will the protection last? Eventually, will the protection work in the wild? And will this allow, ultimately, then the goal would be to be allowed successful reintroductions from survival assurance colonies. Okay, so eventual applications of this might be, as we just saw, we could bio-augment this anti-BD bacteria that could be used to increase resistance of amphibians and then allow reintroductions. Down the road, it may be possible to bio-augment soil, and what I mean by bio-augment is picking good candidate bacteria that are already in the environment, but just increasing their numbers. And we're thinking that if we, for example, bio-augment the soil, that would transfer then to the amphibian skin. So you wouldn't have to do this on a frog-by-frog basis. So this might have the potential to slow or stop epidemics of these waves of epidemics that Karen talked about and allow recolonization, although there would have to be a lot of testing done to make sure that this bio-augmentation didn't have any unintended secondary consequences. However, this bio-augmentation of beneficial bacteria is done widely in agriculture already. You can stop at a garden store on the way home and buy a product like this, which you can see listed right on the label. It has about 15 different beneficial antifungal bacterial species which can colonize plant roots and keep the plant roots free of pathogenic fungi. So I just wanted to briefly thank all the various students and collaborators and funding agencies, of course. There's JMU written out in our bacterial species. Thank you.
Thank you, Reid. We have one more brief presentation. This time it's my pleasure to introduce Dr. Brian Gratwick. He's a conservation biologist. He works here at the National Zoo. When John Berry told me that we were going to launch this initiative or expressed his interest in doing so, I said, John, we don't even have an amphibian biologist. And he said, well, we're going to get one. And I have someone in mind. And so it turned out to be Brian Gratwick, and he's done a fantastic job. So Brian's job is really to coordinate the Smithsonian-wide effort to help halt the amphibian decline. He's working together in a pan-institutional way to recruit people to the cause, and he's been instrumental over the last year in helping to establish the Panama Amphibian Rescue and Conservation Project. Welcome, Brian.
Thanks very much, Steve. So you've already heard a lot about this situation. Amphibians are in big trouble. This is a global crisis. And when I was hired, I was kind of shocked to learn that I was probably one of a handful of amphibian conservationists working full-time on mitigating threats to amphibians. It's my job to try and save amphibians. So when I became more appraised of the situation and realized how few people we have on the planet to actually do something about this compared to other vertebrate groups like birds or even reptiles, we just don't have the capacity to be dealing with this situation. When you look at the incredible species that are going extinct right before our eyes, these species are going extinct all over the world. The gastric brooding frog went extinct in the early 1980s in Australia. Both known species of this incredible frog are extinct in the wild. This photo shows a little baby frog hopping out of its mother's mouth. Now the mother would actually take her eggs up, swallow them, and brood them in her stomach. And when the babies were were fully developed, they would just hop out of the mother's mouth and go on. Now these frogs also produced a kind of chemical in their stomach called a prostaglandin that would line the stomach and prevent the gastric acid from being secreted and digesting these eggs. And that was potentially, a cure for peptic ulcers, but unfortunately, that option is no longer on the table. We have lost the option values from that particular species. Incredible Monteverde golden toad went extinct inside a protected area. The Puerto Rican golden Coqui frog went extinct again, some of its range inside a protected area. These frogs have all been lost from areas where they should be safe. And it's just incredible that it's happening all across the world, from the Caribbean to Australia to the neotropics. Even here in the United States, the Wyoming toad is now extinct in the wild. We've worked out retrospectively that BD had a big role in it, and I'd like to recognize Bob Chastain and the Cheyenne Mountain Zoo, who's actively breeding these frogs. If it weren't for the captive assurance colonies that were developed once they realized these guys were blinking out, we would have lost that species. Similarly, Panamanian golden frogs, through Project Golden Frog in 2005, decided that, based on Karen Lips's predictions, that this incredible Panamanian animal, one of the most charismatic animals in Panama, a very important cultural symbol, was in danger of extinction. So zoos took that animal into captivity, and it looks like that species now may be extinct in the wild. So what we're really looking at, you've seen this picture before of the mountain yellow-legged frogs, but when this chytrid fungus sweeps through Central America, you get massive amphibian deaths. And so I went down to see what Smithsonian could be doing, and I went and I spoke to my colleagues at the Smithsonian Tropical Research Institute and at other zoos, and they said, look, we've seen a lot of amphibians go extinct in Western Panama. We've managed to take some limited actions to save species in Western Panama, but there's no one there. Everyone's looking to somebody else to take on the responsibility of trying to save the frogs from Eastern Panama. We recognize that this is our last chance. In the neotropics, in the mountainous neotropics, this is our last chance to preserve the unique amphibian biodiversity. When you think of tropical, incredible biodiverse frogs, you think of the rainforest. Well, that is the last place on the planet that we actually have a chance and a responsibility to do something about it. So back to the story in Western Panama. I'd like to recognize Houston Zoo here, who realized that as this fungus hit, that zoos really needed to be involved, and they took up the challenge and started literally just grabbing frogs as they were dying, as this fungus had hit in the mountainous regions of El Valle, and started packing these frogs in cages and actually stuck them in a hotel room because they had nowhere to put them initially, and then eventually built this new center called the El Valle Amphibian Research and Conservation Center. And they now have about 17 species there from Western Panama that are critically endangered. And they're breeding. This is Heidi Griffith, one of the center directors down there, showing Steve Montfort an incredible collection of frogs. These are some of the species that they did manage to pull out of the wild. You can see Panamanian golden frogs at the National Zoo. In fact, we have some here today that you could look at. We also have Hylomantis lemur in an exhibit we could show you afterwards. But you have these just incredible, diverse, beautiful frogs that we were lucky we managed to get into captivity. But that begs the question, what about eastern Panama? There are incredible, diverse range of animals, and I'm just going to show you a few images. This is Atelopus certus. It only occurs in a very, very small single mountain in the Darien region of eastern Panama. And you know, when we know from what's happened to other Atelopus species around the world, that this one probably will go extinct very quickly as soon as BD arrives. And there's no captive assurance colonies of this amphibian in captivity. This is just some of the beautiful biodiversity that we stand to lose. And a lot of it in these regions hasn't been, that region of eastern Panama has been quite poorly explored herpetologically. Some places haven't been visited since the 1960s. So with new technology to describe these species, we think there's probably going to be a whole lot of new species. So an assessment done by Amphibian Arc earlier on in this year discovered that they did an assessment of all known species and estimated about 25 species from eastern Panama will probably lose, and we need to establish some assurance colonies. So that's a big problem, and that problem is far too big for any one institution to tackle alone. And you know, everyone is kind of daunted by the size and scale of what you've got to take on, so everyone kind of looking around at everybody else, trying to see who's going to be naive enough to really try and take this on. But we can do it together, and I think that this incredible project, and I just want to thank our partners right here, right now, because without being able to leverage other people's skills, the Cheyenne Mountain Zoo has a lot of experience with captive amphibian research. The Zoo New England, with John Lenihan here today, is providing the lead veterinary care for establishing biosecurity protocols in a new facility we're planning to build. Biff Birmingham from STRI is helping us to convene other scientists to be starting to think systematically about answering some of the science questions. Defenders of Wildlife, I'd like to recognize Jamie Clark here, the Vice President of Defenders of Wildlife, who is going to be helping us with the public outreach, educating people in the United States. And I want to thank you here today, because getting this message out is one of the most crucial things we need to really mobilize our cause. And I'd like to recognize Africam Safari. Amy Camacho is the director of the Latin American Amphibian Specialist Group, and so they've had a terrible time with this disease in Mexico, and they have some captive breeding propagation facilities going right now. And Houston Zoo started the rescues in Western Panama, and they'll be providing us some technical assistance in Eastern Panama and help us to learn from their lessons dealing with Western Panama. And we'll try and be building up some capacity to do that. And all of this will be taking place at the Summit Zoo. It is a city-run zoo in Panama, about 20 minutes from Panama City. The director, Adrian Benedetti, has been absolutely instrumental in helping to facilitate this and helping us to help Panama build its capacity to deal with this crisis. I think it's very important that we now sort of move away from this model of, oh, well, we'll take your biodiversity and we'll look after it. Instead, we're saying, hey, we want to try and build some capacity to actually manage this crisis right where it is. So this is a photo of exactly how far we've got. It's a modified shipping container, and we plan to have over the next 3 years 6 modified shipping containers. They're basically just shipping containers that have air conditioning, climate control, water, and they're just lined with hundreds and hundreds of tanks that will be used to house critically endangered frogs that we target from the field. And when we go and start targeting these species, we have to realize, I'm just going to go back a bit here. When we target these species, it's okay. When we target these species, we're faced with a really daunting task. If we pick the wrong species and a different one to establish a captive breeding program for, and a different one goes extinct, then we've really missed out. We've lost an incredible piece of biodiversity. So we have to maximize our probability of actually mitigating an extinction by successfully predicting which ones will go extinct. And thanks to Karen's work, we have a fair idea of which ones those will be based on the previous witnessing the fungus sweeping through. And we need to maximize our knowledge of how to keep these things in captivity, which is why our zoo partners are going to be so instrumental in developing husbandry techniques. Some of these species that are priorities, we don't even know how they breed. We don't know if they lay their eggs in water or if they have direct development and just lay it in moist leaf litter. So it's just an incredible opportunity to actually do something. So our strategy is to establish a true amphibian arc, something where we can buy some time to develop a cure. But we cannot really justify going and setting up all of these assurance colonies and hoping that somebody else comes up with a miracle and develops the cure, which is why I'm so excited about working with Reid Harris and Louise Rollins Smith, because I think that this probably is the most exciting piece of news that I've heard about being able to do something about the BD question. There's a lot of questions we do have to answer, but if we sit back and wait 10 years until that gradually takes its course, we will have lost a lot more. So this project, through our partners, is probably now one of the largest amphibian conservation initiatives in the world today that's actually doing something to mitigate threats and stop amphibians going extinct. So I encourage you to look for more information on our website, amphibianrescue.com, and to help us save amphibians. Thank you.
Well, thank you, Brian. I'm going to invite all of the speakers to come up front, and then we're going to entertain some questions from members of the media. And then also, if you're interested in having an interview, our public affairs staff is in the back, and they can set up an interview for you. And then after that, finally, we've put some frogs on display behind us in our exhibit that we're currently renovating, so you'll have an opportunity to do that. So could you all please come up here for just a few minutes? Maybe stand over here. Are there any, also all of the partners will be available as well for interviews or questions. So can we go ahead and we'll take some questions now. Here.
I'm wondering if anyone has been able to identify the possible origin of the fungus?
Okay, the question was about, has anyone been able to identify the possible origin of the fungus?
One of the theories that I think is probably got the most weight right now is that this fungus originated in southern Africa, and no, it's contentious. But one of the theories is that it originated in southern Africa and has been spread around the world on African clawed frogs that were being used for pregnancy testing in the 1940s and 50s, which might explain how it got around the world and has showed up in all of these different places. But there are other ways that this fungus can be transmitted. People have thought that maybe amphibian researchers themselves might have just inadvertently, from going from different place to different place with their equipment, might have played a role. There might have been a role with the international trade in amphibians for food. There's a lot of different competing hypotheses.
Other questions? Back here? Yep. The question was, even if you can reintroduce the animals that have a cure to chytrid, how do you prevent other sorts of human-induced loss of habitat and loss of biodiversity?
Sorry. The thing about chytrid fungus is this is something that's happening very rapidly. Within 5 months of arriving at a site, you will lose these things. So it's almost a catastrophic event. Things like climate change are gradual. There is some evidence from Madagascar that amphibians are being forced up their mountain slopes from gradually warming climates. But I think that this is an immediate problem. There are people, you may have heard, working on the climate change issues, so we're just hoping to buy ourselves some time to deal with some of these other things. There's also some great work being done to preserve habitats in the tropics.
Any other questions? Okay, well, our guests are going to stay around for a while. If you'd like to come up and ask questions in person, please do that, or to visit with any of our partners. And please feel free to come and see our frogs that we brought here for you on display, and some of our staff can kind of talk you through that. Thank you all very much for coming.
We have to consider all sorts of problems, but this is happening.